this afternoon, and I'd like to call Mike Kolios up to give an introduction of the day. So thank you.
Thank you, Ori. My name is Michael Kolios, and I'm the Associate Dean of Research and Graduate Studies in the Faculty of Science at Ryerson University, and also one of the co-directors of the Scientific Operations Committee of iBEST. It's always a tough act to be after Dr. Ori Rothstein. I mean, trying to best them, especially on iBEST Day, is a pretty tall task. So I'll talk a little bit about what we're going to see today in the presentations. And of course, our first event at 9:30, and I think we'll be on time, is the keynote speaker, where Dr. Ali Khademhosseini will be talking about his work on nano and microfabricated hydrogels. And I'll be introducing him right after my little overview. Then we have, the way we've structured it is we have in the morning what we would call a trainee event, and in the afternoon we have a faculty event. So in the morning trainee event, we have this special session. And we've realized that we need to prepare our trainees for a wide variety of career paths that are available to them, either in industry, academia, private practice, government, hospital sector, etc. And many times, the career path is uncertain, it's unclear. So career exploration is actually very critical, and having trainees talk to individuals that have traversed those paths is very important. So in the spirit of that, we have this trainee event called Industry Insights, where we have collected a variety of industry professionals, and they will be talking about their career paths themselves. And I would very highly encourage the graduate students and the trainees to talk to them either at the poster session or right after this event. It is very important for you to see how other individuals were able to navigate these different paths. After that, we're going to have our scientific exposition. The most important, of course, part of that is the lunch, but together with the lunch, you will have posters and displays from our community partners, our scientists and staff from both institutions. This is an excellent opportunity to intermingle, to bounce ideas, and to spark collaborations. And indeed, some of the collaborations that have been initiated were from exactly events like these, from developing models of concussion using high-intensity focused ultrasound, to using photoacoustics, to look at the suitability of organs for transplantation. A lot of those ideas were first initiated in discussions in these kind of poster sessions. So I'd highly recommend you interact with all the individuals. So after that, from 2 o'clock to 3:30, we will go to our faculty event, which is the spotlight on the research themes. As Ori mentioned, we have these 4 research themes, and the panel, as you can see at the bottom here, has individuals that are the theme leads for all of those themes, other than the biomedical tissue and engineering repair. Haibo unfortunately could not make it today, so there's a replacement for him. And there'll be presentations from individuals who do research that is related to those themes, and then the panelists will be asking questions to try to see what kind of collaborative activities we could promote in these themes. This session will be chaired by Dr. Stephen Waldman. So right after the research theme, we'll go to our last and second keynote speaker, Dr. Rita Kandel, who is a chair and professor of laboratory medicine and pathobiology at the University of Toronto and chief of the Department of Pathology and Laboratory Medicine at Sinai Health System. The session will be chaired by Dr. Yenny Yücel, a very active member of the iBEST, and we'll be closing the event after that with some closing remarks from the Dean of Engineering and Architectural Science at Ryerson University, Dr. Thomas Duver. So what I want to do now is introduce our keynote speaker, and if I were to summarize, it would just be wow. So let's see. Dr. Ali Khademhosseini received his PhD in bioengineering from the MIT in 2005, and his MAC and BAAC from the University of Toronto. both in chemical engineering. So we have a Toronto connection here, and we're very happy to host him. He's professor of medicine at Harvard Medical School and director of Biomaterials Innovation Research Center at Brigham and Women's Hospital. He's also faculty at the Harvard MIT Division of Health Sciences and Technology, as well as associate faculty at the Wyss Institute of Biologically Inspired Engineering. He's recognized a leader in combining the micro and nano engineering approaches with advanced biomaterials for regenerative medicine. His laboratory has pioneered numerous technologies and materials for controlling architecture and function of vascularized engineered tissues. He has authored over 500 publications. His H-index is over 90. It's rare for me to meet somebody who has an H-index that's double their age, but anyway. And he has over 30,000 citations. In addition, he's delivered many, over 300 invited and keynote lectures, one of which is this one. He's been recognized by over 40 major national and international awards, and I would like just to highlight the Presidential Early Career Award for Scientists and Engineers, the highest honor given by the US government for early career investigators. He is fellow of many societies, which you could see in the handouts that we've given to you, and he currently serves as an editorial board member on numerous leading journals in the field, and he's associate editor for a very high-profile journal in the field, ACS Nano. And he's a member of the NIH Bioengineering, Technology, and Surgical Sciences Study Section of the National Institutes of Health. We're greatly looking forward to an exciting talk by Dr. Khademhosseini in his never-ending quest to recreate and engineer tissue complexity for applications in stem cell bioengineering and tissue regeneration. Ali?
All right. Thank you for that very kind introduction. Not sure if I deserve it, but I'll definitely take it. It's a great honor to come here, actually. Growing up in Toronto, I've always been, I love to come back and see my family and see all the amazing things that are happening here. And I think this is actually a very interesting place because Ryerson, when I was here, it was a totally different school. So the amount of transformation that it has undergone in the past few years is just incredible. And a lot of it goes to the incredible visionary leadership, the great partnerships with the hospital here, as well as great faculty and incredible students. So I'm really excited to be here. And I actually, very thankful to my hosts, including Michael, who kind of took good care of me last night. So, I had a easy time sleeping. And actually, this is last night, but I went back in my photos, and I was here 2008. So if you kind of focus here, you may see some. There we go. You may see some faces that you may recognize. So I'll see if you see the differences. I think that I haven't aged too much since 2008, and people haven't aged, but sometimes facial hair gets in the way. So, so I'm going to tell you a little bit about my journey. And as it was mentioned, I actually grew up here. I actually went to Jarvis Collegiate, which is kind of down the street here. And before that, I was at Jesse Ketchum, which is another school, for elementary school. And so I've been in Toronto for many years, and I started thinking about what I wanted to do for university. So I was good in chemistry, so I did my OACs. Back in the day, they had them. And then I'm like, can you find a job as a chemist? They're like, yes, but better if you kind of add some engineering. So I went to chemical engineering, just kind of not knowing what it was about. And when I went there, I really learned, I appreciated how engineers can solve problems by simplifying the complexity in systems, and then being able to just try to make the right kind of assumptions to solve these things. So I'm like, okay, great. So I have some problem-solving skills. And I started thinking, what can I apply it to? So then after my third year, I went to work as an undergrad with Michael Sefton, who's a professor in chemical engineering at U of T. And that really changed how I thought about things. I started thinking about research as this really incredible way of solving real world problems. I started actually working on regenerative medicine from the beginning, and I just fell in love with it. So I did my undergrad and I did my master's, and then I was lucky enough to go to MIT, and that's just been an incredible journey, and I've just been very grateful for everything. So what I'm going to talk about today is part of the research that we do, and it's very much analogous to the vision that was outlined earlier, where I'm an engineer, but I try to solve medical problems. But as an engineer, I only know so much, right? So this partnership of engineering and medicine becomes particularly important. So I'm going to highlight that a bunch in my talk. And as an engineer who works at a hospital, I actually get exposed to a lot of these medical problems that drive our research. What is one of the things that we do is we try to engineer materials. And this picture kind of shows some of the ways in which we think about things. So we basically try to engineer materials the same way that nature builds complexity. So when you think about nature, of course, everything is very well organized. And at different length scale, this organization is maintained, and it leads to the right kind of functionality that biological systems have. And this incredible organization at different length scale is what we also try to do in some of the materials that we develop. So we often do start off with the right kind of biological molecules that we can engineer. We can use genetic engineering tools to kind of make the right kind of proteins or polysaccharides or other kinds of things that we want to use as the basis of our material. Then we use things like chemistry to be able to modify these materials even better, to cross-link them in particular ways, to make them so that they actually have the right kind of features that we want. Then we can use engineering at larger length scales to be able to control the architecture of these things, because our own tissues are very much controlled in how the architecture of these cells and tissues are built, and how that leads to functionality. And finally, we try to actually use this in medical applications. So if I had to kind of break this down into the kind of materials that we do, we work a lot on gels, things like Jello, which everyone is familiar with. And we try to make better Jellos that can be used for making tissues. And this sounds funny, but it's actually true, because Jello is made from gelatin, and we do use a lot of gelatin-based materials in our systems too. And the good thing about Jello is that it's cheap, so I can give it to my students without having to worry about if I'm going to break the bank. So we make these gels, and we try to control their properties. So we play around with different types of features. We can use light, temperature, pH, and other ways to cross-link them. We can use interpenetrating networks and add nanomaterials to them to control various properties of that these materials have. And once we have the right kind of material that we're happy that can affect the cells in a particular way and make potentially a tissue, then we play around with the engineering of these. And we've done a lot of work in trying to build different kinds of engineering platforms to do that, whether it's using like lithographic approaches to control the architecture of these gels and cells into particular things, or use 3D printing and microfluidics. And I'm going to just give some examples of that today. So my work, once we have the right kind of engineering and materials tool set, we actually try to apply it to these different medical problems. And being at the hospital is incredible, because you get to see the real world problems and interact with patients. And this is what I think iBEST is particularly suited to do as well. So we play around with these materials in cardiovascular therapies, with colleagues in surgery to try to make better materials for surgery, direct cells and stem cells to be able to make tissues, and make advances in fabrication, tissue engineering, and this area that I'm going to talk about called organs on a chip, which is a very interesting attempt at trying to overcome the barriers that exist right now in pharmaceutical industry and drug discovery problem. Now, what's common across all of these, and I think what has made our research kind of more fun and interesting, is that we believe in this concept of team-based science where we work in groups and we have people from different backgrounds who work together to solve problems. Now, the challenge when you have these kinds of approaches is that how does the person get the credit? How do you publish your first author papers and get your positions? But my approach to this is that one is let's solve problems, and if you do that, your pie is big enough that everyone can eat. So typically we wound up being having enough output and enough success that these integrated teams of people from different disciplines wound up being successful. So I'm going to talk about a couple of stories. I thought maybe as an example of how our research works, I'll give one example of how myself as an engineer and a clinician teamed up to solve a medical problem, and how it came from the problem to the kind of approach that was mentioned in vitro, ideas of how you can solve this problem, and then pushing it through the translational pipeline, how we can go small animal models, large animal models, and then actually trying to push it through clinical trials. So this is a fairly recent story that was published recently. This is actually the journal cover. And this kind of highlights the kind of approach we're trying to do. We're trying to make a material that can plug a blood vessel, and these procedures called embolization. And this plugging the blood vessel, we're going to show why you want to do that, but the material is made from nanoparticles here and a polymer system. What this does is that it actually makes materials that kind of can be delivered through catheters and can maintain their structure. So this kind of feels like a toothpaste. So you can see that it can be, when you pressurize it, it goes through. And our idea was that we can use this to solve some medical problems. So this problem was introduced to me by one of my colleagues. So this is the picture of my friend and I. This is Rami Oklu. He's now a physician in Mayo Clinic. But what he's an interventional radiologist. So what these people do is fairly incredible. So these guys can go around inside blood vessels and take a catheter and then be able to solve all kinds of problems. So these are, I kind of tell him that he's a high-paid plumber because he can go around and unclog or clog blood vessels. And I know that he's a very high-paid plumber. So, so one of the problems that he does, for example, is he tries to address issues like aneurysms. So these are weakening of the blood vessel walls that cause this ballooning. And a standard therapy is that you can fill these with metallic coils, as I've shown here. Now, these metallic coils come in all sizes of shapes and sizes, and they're very specialized. And the idea here is that they're porous, but what they do is that they decrease the blood flow in a way that it forms a blood clot. So if you put it inside an aneurysm, it forms a blood clot, and it basically excludes this part from the blood vessel, prevents it from rupturing, causing internal bleeding and death. Now, there's other uses here. So here, Rami has actually, this is the catheter, and you can see that he's put many of these coils inside the blood vessel. And why he wants to do that is that because there's internal bleeding. So he wants to plug that blood vessel so that this internal bleeding doesn't happen. Now, this kind of standard therapy has been around for a long time, but there's a bunch of cases where it doesn't work. So why would it not work? Well, in many cases where, especially the older population is on anticoagulant medication, this ability to actually form blood clots inside these blood vessels is hindered. So it doesn't work, for example, in this case, where there's an internal bleeding. My colleague here has filled this blood vessel with coils, but when you put contrast agent upstream, you basically see that it goes right through and the blood does not coagulate. So in this case, this internal bleeding, as shown at the bottom, would continue, and either you have to really get invasive surgical approaches, and there's many cases where these kinds of patients can't make it. So our approach was, let's come up with a solution to this. As a material scientist and an engineer, we're like, okay, let's think about what the properties it is that you need, what's your design criteria, So we came up with a material that I've shown here, which is basically a polymer made from gelatin and this nanoclay material combined together to make this shear thinning material, which is basically like toothpaste. When you pressurize it, it flows. And why we use this is because these two materials have electrostatic interactions with each other so that they form a bond, electrostatic bonds, that forms this gel. But when you pressurize it, these bonds break and you have a reversible gel. Now, this is a family of materials. It's not just one material, because you can change basically the amount of composition of your gelatin and nanoclay, so the ratio of these two different materials, as well as the concentration of the gel. So what you can then do is you can make these gels and see how they behave. So you can do a lot of analysis. There's a lot of interesting science that you can do here. But what you can see here is that these gels are very tunable. So here, for example, you can see the amount of force you need to actually make these things flow and become liquid-like. And this is the amount of nano material that you have, and this is the concentrations of the material. And you can see that you basically have very tunable properties. You have gels that are very hard and more rigid, and ones that can flow very easily. And these materials kind of are like toothpaste. They're reversible. So if you apply force, this is the basically some measure of mechanics. They become liquid-like. When you remove the force, they go back to the original feature. So this is just shows some of the properties of this. Now, why would you want to use this particular combination as opposed to other things? Well, this is all by design. Gelatin, we know, in the body can actually get degraded and remodeled by the cells. And these nanoparticles are particularly interesting because we know that they can induce blood coagulation. So actually inside the blood vessels and inside the problems that we want, this is a desired property. So here, for example, if you put blood in a dish, it takes a few minutes for it to coagulate. Now, if you have, this is a nomenclature where 9% material and the amount of nano clay here is 0%. So if you just have gelatin, it takes just as long. But if you start adding higher fractions of this nanomaterial, then you get very rapid coagulation. So if you have it inside the blood vessel, then it actually induces this blood clot formation. And these times decrease significantly based on the time. Now, this is an interesting material. So then if you want to actually translate it, how would you go about this? You start looking at things like toxicity, initially in small animal models, and this is the kind of work that we've done. We can put it inside subcutaneously and then see what happens to it. The material here, after a few days, kind of forms a little bit of fibrotic response, like any other material that you put in the body. But after 4 weeks, you see that the material degrades and is infiltrated by the tissue that grows into it, and forms some kind of lower amount of fibrotic tissue. Now, this works a lot better than other things that are out there. This is other hemostat that we tested as a control, and you can see a huge amount of inflammation initially and persistent level of this inflammatory tissue even at 4 weeks. Now, this is good, shows that there's some biocompatibility with the material. Now, would it work in the applications that we want? Again, we can use small animals first. In this case, we can take the blood vessel this is the femoral artery of these rodents, and put the material here to see whether it stays there. Initially, you have a lot of blood flow, and once you put the material, you see the blood flow has ceased. So it works in small animal models. So if we have mice and rats that have aneurysms, we can help them. But what about people? So then you actually need to say, okay, is this material designed in a way that can work in patients? So again, you can start off in a laboratory, make ex vivo models of circulation. In this case, these models can have aneurysms or other things, and you can see in these flow models whether you can fill the aneurysm or you can actually fill the entire blood vessel so that you can prevent things like downstream bleeding. Now, we can do that, and it works through the catheters that clinicians use. And once you do that, you say, okay, now I want to put it in large animals on people. Is it designed properly? So then you can do kinds of tests like, what are the properties inside our own blood vessels, and can this material withstand it? If you put it inside a blood vessel, does it get dislodged and go downstream and cause a heart attack or a stroke or something? So in this case, we can again make ex vivo models where we can take these blood vessels and actually take these pig blood vessels, put the material here, and put pressure back, put flow behind it, and see how much pressure you need to dislodge the material. And basically what you see is that the material, if you have very high pressures, gets dislodged, but these are can easily withstand physiological environments. So if you have these kinds of pressures inside your blood vessel, you have other problems. You don't need to worry about the material dislodging. So once we've done this, then we actually want to push it through the translational pathway. If you want to put it through a regulatory process, in the US, the FDA likes to see large animal data. In this case, pigs are the right model because they have a lot of the same properties as our own blood vessels. So this is my friend Rami actually doing the pig surgeries. And there's a lot of data here. I'm just going to highlight a couple. But the idea here is that, let's say you have this common iliac artery, which is a high flow environment, and one of the larger blood vessels in larger arteries in the body. And we can take the catheter, put it here, and then dislodge the material, put the material here, and actually block and occlude this blood vessel, which is significant. You can do this, of course, if you can do it to arteries. Veins are pretty easy, lower flow environment. But there's other kinds of experiments that one can do. For example, you can see whether pieces of this can get dislodged and cause embolization downstream. So here, you can put this material upstream from the lung, and if parts of this material come out, then it actually goes and occludes regions of the lung. So if you do imaging of the lung, you see that you should see big black areas, and we don't see that in the lung, so the material does not break off and go downstream into the lung, and it maintains the right kind of features that we want. And we've done a lot of short-term, long-term experiments. This is what happens to the material after 4 weeks. In this case, where we see the material still remains there, it occludes the blood vessel, exactly what you want. And what happens is that as the material degrades, as the cells infiltrate the material, they degrade the material, but they fill it with a fibrotic tissue, exactly the kind of behavior that you want and you have right now in these metallic coils. So this is the kind of work that we're doing, and now this has become the basis of startup to translate this to the clinic, and people are interested in trying to push it forward. So that's the kind of idea, but it shows the power of teamwork and interdisciplinary research. You can see things that involve mechanical engineering, chemistry, chemical engineering, imaging, lots of clinical disciplines, and they all come together to solve this medical problem. So this is the kind of stuff that we do. And there's other examples, but for time's sake, I'm going to shift gears a little bit to talk about some of the other things, which also gives you a flavor of how engineering combined with medicine and tissue engineering can solve problems. So the next thing I want to talk about is this problem of drug discovery. As you guys all know, pharma is big business. Making drugs is very costly. It takes many years and billions of dollars to get a drug approved to FDA. And this approval has actually, every year, the number of drugs that get approved has consistently gone down, and the cost has consistently gone up. So the model is not sustainable. The budget for these R&D companies, and we have a whole bunch in Cambridge, is just astronomical. This is actually a couple buildings right from where my lab is, so you can just see these nice buildings go up and lots of investment. Now the big problem, why does it cost so much? Well, the big problem is that when you have these, when you look at the cause of failure, a huge fraction is due to a lack of efficacy and safety of these compounds, particularly at later stages of approval process. So what happens is that these companies typically test their molecules first on tissue culture dishes, then they test them in animals, then they go phase 1, 2, 3 clinical trials where they increase the number of people that are that they test, and they try to see whether it's safe and then efficacious. And the problem is that this model, particularly looking at these tissue culture dishes in animals, do not replicate the complexity of human tissues. So oftentimes they get to phase 3 clinical trial where there's already hundreds of millions of dollars of investment to find some kind of liver toxicity, cardiac toxicity, things that just kills the program. So because of this, there's an emerging theme in trying to actually solve this problem, where the idea is we've been working on tissue engineering of human tissues for many years. Can we actually use this for this drug discovery process? But not just take tissue engineering and test drug on individual human tissues, but be able to actually mimic the entire human physiology on a chip. So we can try to make different tissue types, we can link them together to actually be able to recreate the human behavior, human complexity with respect to how it interacts with these compounds. So that's the idea. Now, there's already been a lot of push in trying to make this happen. So where we and other people are trying to take these 2D monolayers of animal cells that are just individual cell or tissue types and transform them in 3D organ constructs that are made from human cells. So with the human stem cell technology, you can use iPS cells, and you can even use patients' own cells to be able to do this, and uses the microfluidics to link these together and look at the complexity. So when you look at this, there's many different tissues that have already been attempted at being miniaturized. And these tissues, the common themes that you see across all of them is that they try to appreciate the complexity and the microarchitecture of human tissues and use this appreciation with the right cells to start getting functionality that is representative of how human tissues behave. Now, we've been working on this for a few years. This is one of the systems that we've developed where this is actually the hardware, where this is a portable, it's a small incubator where the tissues are housed. And the surrounding components here are basically what you need to be able to monitor how these tissues are behaving, to control the fluid flows, and run the system. But the main components are inside here. So if you zoom in here, we've made our organs on a chip systems in a way that mimics how computers were made when I was a grad student or undergrad at U of T, where I used to go on Spadina and there was all these computer shops. And I had a computer where there was a breadboard and I would put in different chips to make a better graphic and play better games and all these other things, right? So this is the same kind of design. So you can have a breadboard, in this case a little module that controls how the fluids flow, and then you can plug in different components as needed to be able to ask different questions. And these components include things like different types of tissues, includes kind of fluidic components, and includes a lot of sensing components where you can actually monitor things like how are these cells behaving, what is the system, whether it's functioning properly, all in as non-invasive approach as possible. So this is, when you zoom in, this is some of our early versions. You can see the breadboard and some of the components attached. And what's shown here are some of the fluidics. So you can actually see microfluidics here. You can see here, for example, these liquids changing color in real time based on which channels are open and flowing. So there's a lot of complexity and control that you can have in these systems. So with the tissues, you can actually put them in simple reactors if you want. This is a little resealable reactor, little chamber that we can put the tissues inside. And we can take our tissues, whether initially we can make them into spheroids or organoids and then deposit them into these reactors, or we can use techniques like patterning of different sorts, and we can do different tissues. We can put liver, cardiac, and a variety of other tissues. So when we look at these kinds of tissues, all right, let's see, this is, okay. When we look at these tissues, we started off looking at using actually cells that are easy to get. So these are some liver cell lines where you can 3D print them into these reactors and look at the liver functionality. In this case, these cell lines look functional, they express the right kind of markers. And if you look at them over the 4-week period, then they have a lot of the characteristics of what you want from them. They express alpha-1 antitrypsin, albumin, ceruloplasmin over these long periods. So this is good, but these are cell lines, and what we want to do is actually make functional tissues. We require better cells. So we can actually now use better cells to do that. And these are examples of primary human liver tissues, where we can take primary liver cells of different types, put them in the right combination, make these little organoids that have all the right kind of expression of different markers that you want and other things which I haven't explained here. They live fine over our 4-week culture periods. And if you take compounds that were recalled by the FDA due to liver toxicity, and you expose these miniaturized liver tissues to these compounds, then you see the kind of behavior that you want, where you initially, at lower concentrations, you get a lot of green or happier cells, and as you increase the concentrations, you get more of these red or dead or dying cells. So that's good. So there's some correlation here. That's good. So this is one of the tissues. We've worked in a lot of different tissues. This is an example of the heart tissue, where we can take induced pluripotent stem cells that are been differentiated to cardiomyocytes and other support cells. And we can take these, again, we can make these organoids that have the right kind of expression of the right kind of markers associated with these heart cells. They beat, they remain alive. And you can make the organoids, or you can pattern these, and they do the same thing. And you can challenge them again with the same kind of drugs that have been recalled due to heart toxicity, and you can get the same behavior, where at lower concentrations, things are good. As you start increasing, you get the cardiotoxicity that caused these to be recalled by the FDA. So these are some examples of how you can make these individual tissue types. Now, I've been talking about how you can generate more complexity by having these organoids or tissues actually connected to each other and linked to each other. So the way to do that is actually to put them now in the modules of our organs on a chip. So here, we can actually do this system where you can have a heart module, a liver module connected in a loop, so that you can look at the interactions between the heart and the liver. And why would you want to pick these two? Well, of course, liver is very important for drug discovery and drug metabolism. All compounds kind of, tries to metabolize them. And the heart is also particularly important because a lot of the toxicity winds up affecting the cardiac system. So you can do this and start asking simple experiments, right? What happens if you add acetaminophen to this? So everyone knows acetaminophen is toxic to liver. So can you pick that up in these multi-organ systems? Well, if you look at it and you look at the heart component, at different concentrations over time, you don't see the change in acutely in what acetaminophen does to our tissues. But as you start looking at the liver component, as you start increasing concentrations and increasing time, then you start seeing changes. You start seeing more toxicity, and even over short periods of time, you start seeing changes in expression markers like albumin and GSD alpha, which are showing that the liver is being affected, but the heart is not being affected. So that's interesting, but kind of very expected. So can you start asking more complex questions about how these multi-organ tissues and compounds actually interact with them? So this is an interesting example, which I thought actually shows the power of these systems. In this case, the idea was to see whether having the multiple tissue types can actually completely change the behavior of how your tissues respond. So in this case, again, we can have a heart and a liver, and we can add a molecule called epinephrine. What this does is that it actually increases the beating rate of your heart tissue. So this is very well-known behavior. Now, epinephrine interacts with a receptor in the heart tissue, or these heart cells, that also has an antagonist. So when you add the antagonist, it competitively interacts with this receptor, and basically it diminishes the effect of epinephrine, so the beating rate goes down. Now, this is what would happen if you just have the heart tissue present. But what's interesting is that this antagonist is actually quickly metabolized by the liver. So this is what happens in our own body. So when we have the liver module, it actually metabolizes this antagonist and diminishes the effect of what this antagonist would have. So if you just have heart tissue, you get some certain level of beating rate. Once you add the epinephrine, the beating rate increases. If you have the antagonist, the beating rate decreases, and when you have the liver module, it starts going back up again. So these are the kind of things that I think shows the power of this system, and trying to be able to predict things that otherwise will not be possible. And I think down the road, there's a lot of these interesting systems that, based on the question, people can start adapting to their systems, to their questions. But there's a lot of other, actually, pretty interesting ways to think about this and use it. So here, just one quick example, where in this case, everything is kind of automated. So where we have the liver, we have the heart, and we actually have this liver tumor model. So the idea here is, if someone has liver cancer, can we try to pick the right dosage of the liver chemotherapeutic in a way that it kills the liver cells but doesn't necessarily kill the heart cells here. So here we can have this fully integrated model where we can have a bunch of different real-time sensors that can measure the behavior of these tissues, and basically run the system. So the sensors all work fine, but what we can see is that we can add the different amounts of this doxorubicin, the chemotherapeutic, and then see how the liver system is affected. In this case, as expected, albumin would go down, GST alpha would go up, but also at the same time be able to pick up the effects on the heart cell, the side effects, as you will. So you don't want the heart to be affected. And in this case, we can see at which concentrations the heart starts being affected as potential side effects of your chemotherapeutic. So these are the kind of stuff that I think is very interesting. You can think about any kind of testing down the road that you would want to analyze, potentially be done with these systems. You can make disease models, you can have this toxic testing, and other kinds of things that I think that are very interesting. So in the last little bit, I want to talk more engineering, because I know there's engineers in the crowd who are kind of reading some of our papers about technology development, and they want to see some of our latest work. So I thought maybe it's good to talk about some of our 3D printing efforts, because that's a very kind of hot field right now. There's a lot of people working on it, and I know there's interest in the community. So one of the things that we've been trying to do is to try, for many years, to try to control tissue microarchitecture, right? So, and the reason for that is that if you look at tissues, basically tissues, when you zoom in, everything is very well organized. So I was exposed to this way back when I was an undergrad, where Michael Sefton was working on this modular tissue design, where the idea is that if you zoom in, there's tissue modules, little functional units that have all the properties of the tissue, but they're small versions of it. They have the right metabolic or mechanical properties, but they're small versions of the tissue, and tissues basically repeat themselves over and over, these functional units, to make the larger tissue. So this is the concept. So if you're trying to build tissues, appreciation of what these small units look like and how they function is important, and how to engineer them. So we've been working on trying to build this complexity using technologies for many years. So these are some types of approaches that one can use, different kinds of engineering approaches. But today I'm going to talk about some of the 3D bioprinting systems. So the idea with most 3D printing approaches is to actually build this complexity by building these layers and build nozzle systems that can deliver cells and gels. So here's some examples of commercially available 3D printers, and we do a lot of designing our own, but the typical idea is that you have a nozzle, you suck up your cells and the gel precursor, and then you deposit this and cross-link the gel to be able to build structures on top of each other. And why you want to have this kind of fabrication system is that the actual architecture of these nozzle systems can actually affect what your cells do. So if you just have, let's say, cells in a gel, and you look at in two different pieces of this structure, One is just a bulk gel, where the cells basically don't see anything other than infinite boundaries of the material. And the other one is more of a small architecture here, where the cells actually do have boundaries of the material around them. So if you look at this is same cells and the same gel, but because of the architectural differences, in this case the cells start actually remodeling, pulling on their environment, growing, and making it something that doesn't look very organized. But at the same time, if in this system, you can actually have the cells, as they start pulling on their surrounding, they see asymmetry in their mechanical microenvironment, and that provides signals to the cells that actually allows them to elongate, organize, and form more, in this case, more fibrous-like tissue structures. So we can do this kinds of stuff by just simply controlling the architecture. We can also do things like trying to build vasculature. We can, instead of printing cells and gels, we can print sacrificial structures that can actually allow us to build porosity. So we can build these sacrificial structures, pour cells and gels around it, cross-link it, and then get rid of these sacrificial structures to generate pores, in this case, things that look like blood vessels, where we can endothelialize these and then be able to flow liquids through them. And what's important to realize with a lot of these systems is that you basically want to help the cells in their path to make tissues, because cells have been designed for a long, through evolution, to have this capability to start building tissue. So here, what we do is that this 3D printed structures are basically providing some initial cues. So cells initially are printed, but they start remodeling these structures. So these are some earlier structures, but over time you see that these things completely remodel, and the cells kind of take over this tissue formation process. So we've done this with a lot of different tissue types. I'm going to give some example here. This is some cardiac tissue, where we can start building structures that have these heart cells in them, and then we can start actually having these heart cells mature and spontaneously beat on these systems. Now, if you look at these, of course, you can see that the cells are beating, but if you want to see how contractile and how synchronous they are, you can start actually looking at different spots and track them over time. And interestingly enough, you start seeing, at least in these cases, that these have actually synchronized and beat uniformly and beat fairly vigorously. So in culture dishes, if you have these systems, like what you have here, you can actually get fairly robust contraction of these, in this case, cardiac cells. You can get skeletal muscle cells that you can stimulate electrically, and other things. So here you can see these cardiac cells vigorously beating, and they can actually change if you fold them into these 3D structures. They can change their shape. But what's interesting is that if they're actually folded in a way that can pump liquid as they contract, they can actually move themselves in a solution. Now, this video is not working for some reason, but the idea is that this thing can actually move itself through solution. Now, this ability to actually make tissues that actually can move themselves is interesting and has inspired us and other people in trying to use this as a kind of template for making some kind of attempts at biorobotics. So, and 3D printing is a very natural way to push this forward. So you can get inspiration from marine organisms, in this case, stingray. You can 3D print scaffolds that basically are made from different materials with different mechanical properties. So if you actually have the cells on top of these, and the cells contract based on the scaffold's asymmetry in the kind of mechanical properties, then what it does is that it intrinsically bends the structure in a particular way that's kind of pre-designed. So that's the idea, and we can actually do that. We can you can build the structure, we can put the heart cells on these, have them mature, and here we can start building more organized complexity of how these things can beat and move around. So this is kind of got a lot of people excited. This is like the biorobotics aspect of tissue engineering. People are thinking that could be interesting. I say we're still many years away from Westworld for people who watch that show. but it's fairly interesting. So, and this is some other examples that this is actually from our colleagues at Harvard. This one where this inspiration is from the jellyfish, and then these are some engineered versions of this jellyfish that have the heart cells and the materials. And this is a little worm that can move itself as well. This is from Urbana-Champaign colleagues. So those are some general examples. I wanted to, the last few minutes, talk about some innovations in 3D printing that we've been also pursuing. In this case, the idea, maybe I'll just quickly talk about some of this. The idea is, can we make this 3D printing processes that we do more powerful? So I've talked about building rods, and so far in 3D printing rod-like structures, but by integrating, by integrating the right kind of microfluidics upstream, you can start actually printing more complexity. So here you can print these tube-like structures where because of the way the microfluidics stream from this nozzle, upstream from this nozzle is designed, then it prints tube-like structures where you can actually flow liquids through these tubes at will and print these fairly rapidly. And you can think about how this could be used for building vascular-like structures, as shown in the next slide, where you can actually have these 3D printed structures where you can build in these roadways where liquid can flow through. And you can, in the walls of these, you can actually start putting the right kind of cells that can remodel to form the different layers of the vasculature. The other thing that is interesting in 3D printing is that most of the technology so far has been built around printing just one type of material. So if you're trying to build tissues, it's very important to be able to print like many different types of cells and materials at will to be able to generate the complexity that's associated with the tissue. So a few years back, we started thinking about these kinds of adding complexity to our extrusion processes. And this is one example where one can get inspiration from a spider and how it generates its silk and apply it down the road to 3D printing. So what a spider does is, to make silk, is pretty interesting because it's got a number of different glands, and it can kind of mix these glands in different ways to get the spider silk property that it wants. So you can get this inspiration and then build engineered systems that can do the same thing. So here you can have a number of different fluidic channels where each one can have a computer-controlled pneumatic valve, and then be able to open and close these in a way that the extruded material downstream can have the chemistries associated with these in the way that you want. So that's the idea. So just to show you how this is, maybe I'll show some of this. If you have 3 different channels, and you first open one, then the second, then the third, then what gets extruded is basically the chemistries that are in these different channels sequentially. Now, if you have them all open at the same time, then all of these will be expressed in the same region of this extruded material. So this will allow us to have a lot of ability to control the properties of these things. You can think about other additional ways you can do this. For example, if you have the 3 channels again with different materials inside them, and you open them sequentially, but you have regions where multiple channels are open, then this extruded fiber will have not only the chemistries associated with this, but in the regions where you have multiple channels open, then you can actually have changes in topography. You can play with other engineering kind of schemes. You can even have multi-phase systems where you can have air bubbles or oil droplets expressed in these extruded fibers. So that's the idea. So the technology you can have a lot of control over this. And where we're going with some of this approach is, I've been talking a lot about how I work with clinical doctors. So minimally invasive therapeutics is a big direction, and being able to deliver materials in a controlled way through small nozzles in the body is going to be important. So these are some examples where you can actually start now delivering cells where you can have these fibers that in different parts can have different cells, in this case, liver cells, fibroblasts, and other regions where you can have these liver cells in close proximity to these fibroblasts surrounding them, so that you can actually maintain these better, as shown here, because these interactions is vital for maintaining the liver cells. Or you can have these extruded fibers that can release molecules in different parts, so that to control the surrounding biology. So here, these extruded fibers can actually have chemoattractant molecules that tells these neutrophils to go to particular regions here and not other ones. So these are the kind of stuff that I think will greatly help these kinds of minimally invasive regenerative therapeutics in the future. So when it comes to 3D printing, it's a very obvious link to take these kinds of technologies and employ it upstream from the nozzle-based 3D printers to generate complexity in what you print. So here, if you just have two different channels and you can control whether A or B is open, then through this nozzle, you can build structures that have different materials and different layers, or even the same layer can have both of these materials expressed and generate this kind of complexity. And we're now build these fairly sophisticated 3D printers that can have a number of different inks, and it can basically express them individually or sequentially in kind of in the way that you can even mix these to generate a lot of complexity in what you actually wind up printing. So in the same way, through the same nozzle, you can build all these different printers. And why this is interesting is the alternative is to basically change nozzles every time you want to print a different material. And these systems that use an individual nozzle are dramatically faster than multi-nozzle printers, and that will help us significantly in building better tissues. So now we're trying to adapt this to tissue engineering systems, and hopefully in a few years when I come back for iBEST 2014, I will give you a lot of really interesting progress on how this tissue engineering systems can potentially be used for clinical applications. So with that, I wanted to end the talk and but give the young people some parting thoughts. But first, I'll do my acknowledgments, where we have a lot of good people in Boston who do all the work, and I get the benefit of going around and being the traveling salesman. So definitely the lab, we have had a lot of people from Toronto over the years, a lot of Canadians who come and do incredible things. And actually just this morning, I already recruited multiple other people. And as it was mentioned, my best recruitment from Ryerson has been my wife so far. And also funding sources and great collaborators and mentors over the years. And I thought I'll put my parting picture as this one, because I like this picture. It inspired me when I was an undergraduate student at U of T. I attended the Gardner lectures back in like 1998 or 7 or something like that. And one of the awardees presented this, and it really stuck with me. So this is actually a painting, self-portrait by René Magritte, which is a very famous surreal artist. And in this, he's actually looking at the egg and is drawing the bird that's kind of emerging from it. And it's, I think it's very relevant for all kinds of research that we're doing here at iBEST and around the world. And it's good for the young people because they have to kind of see the vision of what it is that they're trying to do. So I think this is a very, something to think about. So with that, I'd be happy to answer any questions, and thank you very much for the invitation.
To thank Dr. Khademhosseini again for an excellent talk, very inspiring. So we have time for questions, and I would ask the individuals that want to ask questions to go to one of the 4 microphones that are in the theater to ask the question, please. So are there any questions for the speaker? Up top.
Thanks a lot for this very exciting talk. I have a question about testing drugs by means of your systems. Sometimes it's not the drug that cures, but the metabolites. of the drug, and how well your system potentially may produce such metabolites?
Yeah, excellent question. So some of that stuff we're just starting to test right now. The idea is that having the complexity of organs like the liver, where you can actually have the drug transform into the metabolites and other things, it's actually going to be more predictive. So we haven't done a full analysis of that, but I think that's the idea, that you can look at those kinds of complexity in these systems down the road.
Thank you very much. A question again about this, I guess the same model, and that is, you look at the human studies, many drugs fail because of idiosyncratic reactions. 1 in 50,000, 1 in 1,000, 1 in whatever, pick the number of humans get this side effect. How do you pick that up here? Like those are all presumably the same cells, the same cell types, going to be identical. What causes the problem is the heterogeneity among humans.
That's an excellent point. So one of the directions that people are thinking about these to make it more relevant for personalized medicine is actually to use them as screening platform for individuals. So you can actually have the cells, you can take the skin cells from the patient and be able to make a human on a chip model of that individual patient and then be able to predict toxicities associated with that. Yeah, so that's, I think, a very interesting way. The jury is still out whether that will be cost effective enough and useful enough in the future, but if someone has some it's a genetic factor that would make it more susceptible, it may make sense to do that.
Are there any more questions from the audience? Actually, I have a couple. So I'm going to start with one question, and this is more kind of philosophical more than anything else. You mentioned that for a lot of the work you're trying to do, and for a lot of the work that we're trying to do here at the iBEST too, you have to have very multidisciplinary teams, right? You have to have physiologists, engineers, medical people, teams that have different cultures, different languages, different incentive structures, etc. So if you were to say what are the 2 or 3 top elements of success in order to be able to weave a team like that together to solve these problems, what would they be?
I think it's not always trivial to build these teams. I think one is being in the right environment where you have the different people with low enough borders for them to interact. My best collaborations have come from working with friends who we just sit around and then we realize through friendship that there's opportunities for building these teams and addressing the problems. I think that's very important. And one of the things is having mutual respect and also mutual understanding that different people bring different expertise. So everyone feels that they're part of a team, but not working for someone else who, may be, higher in the ladder or something like that. I think the other thing that I thought is very interesting, and when I went to MIT, I kind of saw that having proven examples of how these teams or this approach can lead to success. So when I went to MIT, I joined the laboratory of Bob Langer, where basically it's like Noah's Ark. You have a couple people from every different discipline working together, and basically there's no borders because everyone is in the same environment, and you have lots of proven examples of how this approach has generated value, whether that would be companies that have come out or therapies that have spun out. So I think having some of those proven examples just kind of changes the way people think it's possible to do research, and I think that was very beneficial for me.
Okay. So we have time for one more question, so I'll ask you to.
Great lecture. I was looking at the models that you built with the cells contracting together, and obviously it's emergent, I guess, that they work together, but have you looked at why energetically they would contract at the same time, how they are?
Yeah, so, and I think some of that has to do with the maturation level and the kind of the bioreactors and the environments that we put them in. So when we try to build these cardiac tissues, oftentimes we try to put them in the best biological environment that we can make in a dish, but also add stimulations like pacing them electrically so that it helps them in making the right kind of connections and junctions with each other, so that down the road, when they have to beat spontaneously, then they have those built into the structure. So I think if you just put them together, initially they're very asynchronous, and through this maturation process, they organize.
One more question?
For the biological system, they are building, but at the same time, they are able to deconstruct themselves and basically disappear. Okay? So what are the, how this waste management system that actually the removal of the material also is non-invasive?
Yeah, excellent point. So and that kind of removal of whether it's a material that we have in there or the cellular components that undergo apoptosis or some other form of removal, I think is very important. It's definitely underappreciated in the field. Most people really focus on just getting them to build stuff as opposed to right now doing what development does, which is build and remove at the same time. So I think it's underappreciated. Some general approaches that people are trying to, principles that they're trying to use is that to make sure that the materials themselves are cleared from the body, degradable, but can get cleared. And the material is vascularized enough, the tissue is vascularized enough that as the tissue's own macrophages and other kind of cells that engulf things come in, they have access to the cells that would, let's say, die in those systems. But it's not a solved problem yet, and not as many people have worked on it as they should.
Okay, good. So I think we'll wrap up now. I'd like to thank again the speaker for an excellent talk. And as a token of appreciation from all of us at Ryerson University and St. Michael's Hospital, I'd like to give the speaker a token of appreciation. Thank you again very much.
Appreciate it.
So now we're going to have a coffee break, and we're going to start again sharp 10:45, and the coffee is in the back of the auditorium. So please be back at 10:45.
Good stuff.
Excellent talk. Excellent
Yeah,
talk.
thank you.
Questions to ask you.
Thank you.